So if we think about this, these molecules are huge, right? So they're acidic by nature, therefore they're called deoxyribonucleic acid and ribonucleic acid, right? Because they're acid in nature, right? And so the two types are DNA and RNA. Now there are other types of nucleic acid, ATP is a nucleic acid, but we're not gonna talk about it today. We're just gonna simply talk about DNA and RNA. And we're gonna talk about them very high level only, right? If we look at DNA in itself, right, it is responsible for all cellular activity. It directs the production of proteins. It's double-stranded and helical. It's maintained by hydrogen bonds, which hydrogen bonds are really attractions. They're very weak, but a whole bunch of together make something really, really strong, okay? And therefore, they can survive temperatures up to 70 degrees Celsius, high salt concentrations. They can survive in acidic environments, and DNA can remain viable in the environment for years. So people who got sent to prison in the 70s or 80s, scientists are now looking at their cases because we are now at the point with molecular science where you can no longer hide from science. And in some cases, what they're finding out is the information that was there on the murder weapon or in garments that were left over at the scene, right? Or garments that were used in the murder, that the DNA of the individual who is in prison doesn't match up with the DNA that are on those garments, right? And therefore, that really exonerates that individual. It basically says, you didn't commit the crime, oh, but you're in prison. Oh, but now we have to go through all this red tape to get you out, right? And that's a big problem. But it has happened. The way that things were done in the 60s and 70s is very different than the way that things are done nowadays, right? And if you think that there's corruption now, back then there was even a lot more corruption.
It's a lot easier.
Mm-hmm. So, nowadays, you know, it's really about people being understanding of what DNA really is, right? I love to be a juror on a jury panel. The problem is they don't want me. Every time they see my background and my education, I get released very quickly because the defense doesn't want me there. Because I know too much and it's gonna be hard for them to pull one over on me. Because my wife, I think I told you, my wife was the forensic scientist for the state of Texas on the animal side. And her testimonies put a lot of people in jail. But she was very disenchanted with the entire process because when she was asked to go to court, it was never about the truth. It was always about who had the better lawyer because they always wanted to poke holes into her arguments and they wanted her to make a mistake so they could discredit her and therefore give the jury reason to doubt the validity of the data.
So what was that crime?
The problem is they could never make her make a mistake because she was way too smart. And they kept asking her the same question over and over again in different ways. And she said, we just discussed it. And she would just come back all the time. We just discussed this and I just told you this is the way it works, right? But after a while, she just decided that she didn't want to do that anymore. So when she got 20 years under the belt, she looked at me and she said, I'd like to retire. I'm like, do you have 20 years? She goes, yes. I'm like, okay, well, you can retire. So she retired when she was 48, which was 6 years ago. And I thought by now she'd been really bored. But guess what? She gardens at home. She plays with the kitties. And so she's not going back to work. I just decided she's gonna be at home. And that she has earned that right? So, you know, it really is about who has the better lawyer and not really about what the data says. The data says O.J. Simpson is guilty. But the jury did not know what DNA was. And the prosecutors did not do a good job of relaying the information, and then Marcia Clark made a really big mistake, right? So on the other side were these three high-end lawyers. Are you with me? Johnny Cochran, Mr. Kardashian, Kim and those guys' father. and then some other dude, I can't remember his name, I always forget his name. But I can remember watching this, and Marcia Clark wanted to add validity to her argument. So she said, we'd really like Mr. Simpson to try on these gloves because they were found at the scene of the crime, and we believe they belong to him. And if they fit him, then that will prove that he was there, right?
Cinderella.
Huh?
Cinderella.
Yeah. So, Marcia Clark never thought Johnny Cochran and Mr. Kardashian were gonna say yes. And so all that would do was add validity, add strength to her arguments that OJ did it. The problem is Johnny Cochran was too smart. And when Johnny Cochran moved on over and said, let's let her do it, right, to Kardashian said, what? Just believe me, just follow my lead. And you watch those all those videos, you watch Cochran say that. And Mr. Kardashian didn't want to, but Johnny Cochran said, we won't allow her to do that. And Marcia Clark was taken aback by that. And so then the gloves brought over and OJ put them on and they only went up to here because they got wet. It was rainy that night, and when leather gets wet, it shrinks, and it only went up to here, and OJ went like this where it shows the gloves did not fit. And Johnny Cochran quipped, ladies and gentlemen of the jury, if the gloves don't fit, you must acquit. And that's all they remembered. Therefore, OJ went free, even though the DNA evidence said it was him. It's not about the, huh?
She didn't argue that issue.
She was way too over her head.
I thought it was also because he forgot to take like his anti-inflammatory or like arthritis medication or something.
Maybe that played a role in
Yeah,
it,
I
but
think-
they were definitely
Yeah, I remember
very
them being
small.
really small, but I thought maybe he like didn't take his medication. It's very interesting. I don't like Kim Kardashian at all, but I do appreciate like she has studied a background in law and now she's working on exonerating innocent people, and I think that is probably like the best thing she could do with her platform.
And she has a huge platform.
A huge platform, and now she just released a podcast about, I haven't listened to it yet, it's a true crime podcast if anyone likes that, but it's about exonerating this man. But it's very interesting because her dad was kind of on the flip side of that, you know?
All for money.
Yeah, all for money,
Mr. Kardashian made millions of dollars
yeah.
just with his name. Because the O.J. Simpson trial made him famous. And so even as a consultant, they charged, I don't know, $10,000 an hour. Can you imagine? Let's eat some cookies and drink some coffee and talk about this. Okay, $10,000. Jen, if you ever get that rich, run me a couple of $1,000, okay? Good. So RNA is really about protein synthesis. It's about how proteins are made. And there are about 23 different types of RNA. We're only going to talk about 3 types, right? So if we talk about messenger RNA, the job of messenger RNA is to make a copy of DNA in messenger RNA, or I'm sorry, in RNA language. What does that mean, Tay? Make a copy of DNA in RNA language. What does that mean?
Transcription?
For sure it's transcription, whoever said it over here, thank you for saying that. But what does that mean? Not replication, it's just a small fragment. When you talk about replication, it's about everything, okay?
It's almost like a clone.
Okay, a clone, but there's a difference in RNA. What's the difference?
The single strand?
They split into
It does have a single strand. You're not incorrect.
But it's the.
You're correct when you say that.
It has 2 strands and then enclose the other.
That's
The other
replication.
one.
We're talking about RNA. Huh? So the difference between RNA and DNA is the types of nucleotides. And in DNA, there are 4 nucleotides. They're adenine, thymine, guanosine, and cytosine. But in RNA, there is no thymine. It gets replaced by uracil. So essentially, messenger RNA makes a copy of DNA, but in RNA language, it puts uracils where thymine should be. Okay? So then, the messenger RNA goes to the side of the cell where the ribosome is at, and it forms a complex with the ribosome. And in that messenger RNA are all of these nucleotides, right? And 3 of those nucleotides equals a codon. What is a codon? Huh? 3 of them. And what does it code for? Yep.
There's a codon to start and stop.
That's true.
For the amino
Absolutely,
acid?
that's true. But what does a codon code for, Tram?
Amino acid.
Oh, okay. Amy?
Wait, so it was not amino acid?
Hmm?
So it was not amino acid?
Oh, I'm sorry. Is that what you said?
Yeah.
I didn't hear that. My bad. It's you go okay. An amino acid. So 3 nucleotides equals 1 codon, which codes for 1 amino acid. Okay? Good. And then our RNA, the ribosome itself, folds all of the amino acids on themselves. So now you have an active protein. Okay? So now you know the functionality of each of the different types of RNA that we're going to talk about. There's a lot of others, right? But those are the ones we're going to talk about. Okay?
Will you say the last one again?
It folds the amino acids so that you have a complex protein. Can I have a okay. So let's, as my little cousin would say, can we pretend? Let's pretend that that is a chain of amino acids. It's not active, right? Because it has no binding sites, yes? It's just a chain of amino acids. When proteins start to be made to function, they get folded on themselves. And so there are 4 different structures of proteins, yes? That's okay. So there are 4 different structures or phases of protein construction, right? So you have your chain, now you have this secondary structure, which has got these folds, it looks like an accordion, those are called beta-pleated sheets. And then you have this kind of curvature that's called an alpha helix, that's secondary structure. The ribosome, once that happens, will fold this on itself. So now the protein looks like this. And at this point, it becomes a tertiary structure and it is active. Yep. Make a fist in the air and put it high. KK. So if, move that ring, it's gonna interfere with my. So if that is the protein and this is the substrate by which it's gonna act upon, if it fits perfectly like this, there's gonna be an enzymatic reaction, something's gonna happen, right? But if that gets denatured some way, and now when the substrate tries to bind to the active site, it doesn't bind, and therefore that protein doesn't work. Each of the little curvatures in the protein is an active or a binding site. And we also call it an epitope, right? Same thing. But this is a very complex structure now. And what distinguishes the functionality of a protein?
It's receptor site?
That's what actually causes the reaction, but what about the functionality? What establishes its functionality?
The shape.
Shape.
Shape. Who said that? Shape. It's the shape. So if you alter the shape at all, you interfere with the functionality. Okay? Questions? Okay. So if we look then at messenger RNA's functionality, it's transcription. If I'm talking and you guys are taking notes, you're just transcribing notes. You're taking the information that I am talking about, the story I'm telling, and you're putting it into your own story in your own words, right? Transcription. Translation is looking at the information that is in that messenger RNA, and then putting a sequence of amino acids in a chain. Translation, yes? What do the codons say, and how we put those things in a chain, right? And then the ribosome is what completes the function, or completes the building of that protein. It folds up the structure. Okay? All nucleic acids are constructed of a 5-carbon sugar, which in general is called pentose, but in DNA it is actually deoxyribose, and in RNA it's ribose. It's constructed of also a nitrogenous ring and a phosphate. Okay? So those three things, phosphate, sugar, and a nitrogenous structure that has rings in it, or a ring, is what makes up the DNA or the RNA. And so here are the base types. Here are those nitrogenous rings. There are two types. They are the purines and the pyrimidines. Yes, ma'am.
Can you go back to what in RNA, what the 5-carbon sugar is called? And what it's called?
Ribose.
Ribose. And in DNA it was called?
Deoxyribose.
Okay, I knew that.
Yep. It's right here. Right? So a 5-carbon sugar, one or more phosphate groups, and a base containing nitrogen. Okay? So here are the bases, because we talk about the nucleotides by the name of their base. And their bases, there are two types. They're called the purines and the pyrimidines. The purines have two nitrogenous rings, right? So here you go, listen, 1, 2, 1, 2. And so the two purines in DNA and in RNA are adenine and guanine, right? In the pyrimidines in DNA have one nitrogenous ring, and they're cytosine and thymine. But in RNA, there is no thymine, so it's replaced by uracil, which is also a one-ring structure. Okay? So let's just make sure we understand this. If I see you at HEB, Amy, I'm going to come to you and say, right now, list the two purines, and you're going to say guanine and
No, wait.
adenine. Very good. And I'm going to, if I come up to you and I say, now list the 3 pyrimidines, and you'll say,
Is it the one that is top?
one ring.
One ring and then there's a 2 ring?
So the 2 rings you already told me, adenine and guanine. Now what are the ones? What are the 3 structures that are only have one ring? Thymine,
Uracil?
uracil,
Cytosine.
cytosine. Okay? The rules of binding. So we looked at this very hard. There were some scientists that came way before us, and they all decided in order for the double helix to work, there always had to be a purine attached to a pyrimidine. Yes? Because if you had 2 purines attached, it wouldn't fit inside of the double helix strand. If it was 2 pyrimidines together, it would be too small and it wouldn't go out enough. But if you had a purine and a pyrimidine, it fit the double helix just perfectly. Okay, and the rules are, if you remember the preposition at, you can never make a mistake, because at adenine always binds to thymine. Therefore, guanine has to bind to cytosine. Okay? Because they're very specific in their base pairings. Good? At. Adenine binds to thymine. Therefore, guanine binds to cytosine. Except in RNA, what binds to adenine? Uracil. Good? So here is the structure of DNA. Right? How do I know it's DNA?
This thymine?
It's got thymine, right? Do you notice these little dotted lines between the nitrogenous bases? What are you going to tell me?
Hydrogen bonds, A and T form 2, guanine and cytosine.
You made me very happy today. 1 point for everybody.
What was the question? What was your response?
So the little dotted lines between those are hydrogen bonds. Adenine and thymine are held together by 2. Guanine and cytosine always held together by 3. Okay? I need a model. Eric, come here. Come and stand by me. So if 2 strands are running parallel to each other, drive with me down the street, they move together like this. Okay? Go stand up. DNA doesn't work that way, nor RNA. They are anti-parallel. Come drive toward me. So therefore, the strands go in opposite directions. So thank you, sir. So we talk about the forward direction of DNA replication beginning at the 5' end and ending at the 3' end. We can always tell the 5' end because there's a phosphate group attached to it. We can always tell the 3' end because there's a hydroxyl attached to it. So you can see that this prime is here, and there's a phosphate there. So that's the 5' end. And here is, it doesn't show the hydroxyl, but there'll be a hydroxyl here, right? That's going to be the 3'. Good? So if I was to draw this without all the nucleotides, if I drew a structure, this is the 5' end, it would have a phosphate group attached, and then it'd be, and then at the 3' end, there would be a hydroxyl. Yep. You can always tell the 3' end and the 5' end because of the functional groups that are there. 5' end is going to have what functional group? Michelle?
Phosphate.
Phosphate. 3' end is going to have what functional group, Anayeli? Hydroxyl. Yes, Jim? Okay. Good? Questions? All right, now. The central dogma theory states exactly how DNA works. And there are 2 tenets of the central dogma theory. The first tenet says that DNA is the master molecule and it regulates everything that goes into the cell, including its own replication. Okay? The second tenet of the central dogma theory is under the direction of DNA, RNA produces proteins. Are you with me? That is the foundation of what DNA and RNA do.
Say under the direction of DNA.
Under the direction of DNA, RNA produces proteins. Okay? Good? All right. So, here is a beautiful image showing the central dogma theory. So here you have this bacterial cell. We're going to use bacteria. We're going to use bacteria as the model because in eukaryotic structures, it's mostly the same thing except there's a lot of work that has to happen before proteins are made. But we're not going to talk about eukaryotic cells. We're going to only talk about bacteria because I'm just trying to show you how it works. I don't need you to become experts in DNA and RNA. I just need you to have a very high understanding of how it works. Okay? So here you have one cell with DNA in it. That DNA gets copied, replicated, and each of the new daughter cells that are produced from this one parent cell are going to have an exact copy of the DNA. And that happens with every single cell that undergoes asexual reproduction. Does that happen in our bodies?
Yes.
Yes. Which cells of our body undergo asexual reproduction? All somatic cells. Okay?
Everything except red blood cells and gametes.
Everything with red blood cells and gametes. Okay? Good? And the reason red blood cells are because they're not nucleated, right? Here, you can see, here's the DNA molecule, piece of the DNA molecule come over, and then that RNA makes a protein. This other thing is recombination. Now, recombination has happened in nature forever. But we now have the technology to do it in a laboratory. We can take genes of interest that we like and put them into organisms and make GMOs. But GMOs have been made forever, naturally. Or farmers would do it, or ranchers would do it. Ranchers would have this big, nice animal and they want it to have a lot of progeny, so they would use the sperm for that particular organism to produce a whole bunch of progeny. Yeah, yes, is that,
So you're saying like, when people breed animals, like dogs, say somebody breeds poodles and they like a certain color, and this male dog, they take the sperm from that and put it in the same color of a female dog, and that's the same kind of.
No, that's different. That's just keeping the breed pure. But if I wanted a new animal, what I could do is take a poodle and a Doberman, and I could breed.
And that would be the recombination, okay, okay.
Okay, and that's how new species, well, in dogs, we call them species, but we call them breeds.
So it would have to be interspecies like that.
No, but that's just it. So in order for nature to occur in nature, it has to be the same species. But I can take genes out of, I don't know. Okay.
I just, there's,
Okay.
so, see, scientists have wanted to replicate spider silk, but it takes too long to get that from spiders, so they took the protein gene from the spiders and they put it in goats. And then the goats that, or then get pregnant, and the babies that they produce now carry that protein. And so they can synthesize that to the spider silk.
Well, we can do it even easier now because we've trained yeast to do it. And it happens a lot quicker in yeast. So all of our spider silk, that webbing material can be made from yeast. So we don't have to rely, the goats were the
Well,
the
it
alpha
was just a
side. Yeah,
video
yeah.
that I listened to last
The
night.
goats made it, they grew it like as hair.
No, they grew the protein.
It's just the
inside,
protein,
yeah.
and then they would take that.
So they would take the protein out of the babies from.
But now we can do it in yeast.
Does that make sense?
Because yeast,
Kind
the yeast propagate a lot quicker,
They just
45
took the
minutes.
protein out of the goat, they put it in.
But we can do it with a lot of
To
things.
reproduce it itself.
So GMOs are in HEB all the time, right? Pluots, broccoli flour. There's a bunch of them.
Cotton candy grapes?
Yeah, those. All of those groupings, those are all GMOs.
Wait, what was it,
Pluots, broccoli flour, tangelos,
Seedless watermelon.
seedless watermelons. We took people didn't like the seeds in the watermelon, so they found a gene of interest that didn't produce seeds, and then they put that into the watermelons, and then people could have seedless watermelons. I don't know why people would want seedless watermelons, because I always want the opportunity to get a watermelon from a seed. That would be really great,
It's a
right?
lot easier with kids though.
Yeah, although I've eaten many a watermelon seed and never was hurt by it, so.
I live right outside of Luling,
Yeah.
so.
Oh, yeah, the little watermelon
That's so
spinning
awesome.
contest.
I love Lloyd.
Jen, you need to go there at least once to see it.
We almost moved to Louisville.
Oh, okay.
Oh, really?
So, hey, folks, in my lifetime, and I got 20 good years left, I believe that we're gonna be able to, like Eric needs a new pancreas, we're gonna be able to make Eric a new pancreas. Because of what we can do with molecular science.
Started doing like the ear and.
We've done all kinds of cool things
Yeah.
already. The other thing that's happening in some countries, not this one, they are making true designer babies, right? Now here, if you are a female and wanna have a child, but you don't wanna have to mess with the other guy in that relationship, you could, if you have enough money, you could go to a fertility clinic, you can pick the characteristics that you want. And they will then take some of your ova and some of the sperm from the donor. They usually take 15 or 20, they fertilize the ova, they freeze them, and then when you're ready to have a child, they can either implant them in your womb, or they can have a surrogate do it for you.
Is it just physical characteristics?
Yeah, because you really can't control for intelligence,
Yeah.
yeah.
Which countries are they doing the designer babies?
I'm not gonna say, you can look it up. I don't wanna be negative toward any one country, China, but, really? So, just gotta be careful. Okay, so reverse transcriptase, is the way that certain viruses reproduce themselves, replicate themselves. Remember, it goes DNA, RNA, proteins, right? Here, it goes RNA to DNA, then it makes a protein. So reverse transcriptase is only found in a few viruses. They're called retroviruses. And the two that are important to you are gonna be HIV and feline leukemia virus. There's others, but HIV and feline leukemia virus are the most prevalent that we would come in contact with. If you have a kitty, and the kitty goes out for any reason, they probably are carrying the feline leukemia virus. Some kitties get affected very quickly and die within the first year of their life because they develop all these different diseases. Some of them can carry it to their life, but as they get older, they start to develop multiple foci of cancer. And that's because of feline leukemia virus. Yep.
I have a friend who, she just had to put her cat down because of that. And before it died, it bit her on the hand, and now she's in the hospital.
For not
They said-
feline, he's probably pasteurella.
No, they said it was something, it was something rare that the cat picked up because of the cancers that it had.
It's called pasteurella multasa.
Is that what it would be? And like, she's like she almost went septic and like all this stuff. It's
That's
just
the catch
Very
she's been in
disease.
the hospital.
Huh?
Is that the same as the catch scratch disease or?
That's Bartonella hemseni. That one's not worrisome at all.
Okay.
Pasteurella is a major.
I actually knew someone who had a fire drill like a month ago, brought her cat out without a carrier, the cat scratched her, bit her, ran off, she ended up having to go get surgery, almost lost a finger. It's an indoor cat, like, but just cats carry something.
They just carry it.
All around their own poop.
Litter boxes or just cat litter?
Yeah.
Litter boxes are
The
disgusting.
toilet.
You guys,
Feline leukemia and
HIV are the two big ones. There are others, but those are the two big ones. Okay?
That's reverse transcriptase.
Reverse transcriptase. So on an example, I might ask you, which of the following infectious agents are known to replicate through the use of reverse transcriptase?
HIV and FIV.
And I would probably only put one of those.
Okay, can you write down the second one, please?
Yes.
Feline leukemia virus?
Yes.
Do we need to know the full word or would FIV be okay? FLV?
So retroviruses.
Oh, FLV. Good.
Fifth.
Thank you. Good.
And I'm sorry, this is a really silly question, but this process is it goes from RNA to DNA to protein?
Well, RNA again, then protein.
Okay, RNA again.
Okay. Good? Question?
And you said HIV?
Uh-huh. And feline, I'm gonna use HIV. I'm not gonna use feline. I'm just gonna use HIV.
Easy to struggle.
Thank you.
So there were some, huh?
It's like feeling lagging
Feline lagging? Oh, no, lagging is part of something else.
Okay, I was like,
It's just feline leukemia virus. Just that. Lagging is something else. That's coming up. Okay? Just- oh. Amen. So there was a group of scientists who wanted to add reverse transcriptase to the central dogma theory. We all said that's just not gonna happen. Because it only is in a few infectious agents, viruses, and viruses aren't even alive. Okay? So they got voted out. Yep.
Is that based on the mutation of the virus? It's like, well, where is it derived from? Because it was not like that before, right?
The machinery of the genes that actually say do this.
But how did that happen? Just evolutionary, basically?
Every animal has got viruses that evolve with them.
Yeah.
So HIV, we believe HIV either came to the human population one of three ways. One, it was always in a population that was sequestered, and then that population got out and HIV got out. The other one is it jumped, which is the pervasive thought. It jumped from a rhesus green monkey into the human population and mutated so that it would infect humans. Right, that's the pervasive thought. Yeah.
Do you really think it was in the way that they think it was, or could it have been a different way?
Like
Sex
the
with
monkey.
monkeys.
Like what?
Would it have had to been sex, or could it have been a different way for the monkeys to the humans?
Oh, it could have been.
They could have
it
bit
been-
'em or
It could have been
bite.
bitten or blood. They were, you know, a lot of individuals in other countries, they eat monkeys, and it could have been through that process of field dressing a monkey.
And it just didn't go right, and,
Okay.
why do they say that it was through,
Why do they say what?
that it was through sex, that somebody?
because it's the most common way.
That's more titillating than
yeah.
saying that there's just animals living close to humans. Sex cells.
Titillating. That's such a funny word.
It was just easier to understand that way versus the other ways. Is
So-
that how it's transmitted?
Yeah.
Let's talk, go ahead.
I'm not
Let's
even-
talk about how DNA replication occurs. So DNA replication really occurs because of the way enzymes work. So you can see that there are two strands of the DNA molecule. Eric, I need you again. So the two strands, just stay right there for right now. The two strands of DNA, one of them is known as a leading strand because it's going in the forward direction of DNA replication. Therefore, DNA gets replicated continuously. The other strand, walk a little bit. The other strand has to get cut, come over, has to get cut, keep going. Then get cut again, then get cut again, and has to be copied, copied, cut, copy, cut, copy, cut, and then put back together again. Okay? That's called a lagging strand. Okay? So in the leading strand, if you look at my enzymes that are found on both strands, you see it's topoisomerase, helicase, primase, and polymerase. But on the lagging strand, there are two additional enzymes that are at play. One of them is called a nuclease. A nuclease is a molecular scissor, it cuts the DNA. And the other one is a ligase. Ligase puts it back together again. Are you with me? So if you ligate something, hold your hand out, Amy. If you ligate something, you bind them together. That's ligate. Okay? I need you again. I need you again, Eric. So, DNA, come here. DNA, do this. Okay, look at everybody. DNA is typically wound up. Are you with me? When it's about to replicate, it will start to unravel, but it's all tensed up because it's been wrapped up like this forever. Are you with me? So as it starts to unravel, it wants to go back together. Are you with me? So there's an enzyme that shows up, and the enzyme hits it and goes, relax, 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 relax. Okay?
That's the Pro V
Okay.
treatment right
Yeah,
there.
yeah, okay, now, hold up. So then another enzyme shows up and it goes, go up, open up. And it opens it up all the way, are you with me? That's called helicase. Don't go, stay open. All right, now there's two strands, one of them leading, one of them lagging, yes? Now the lagging strand gets copied how? Continuously, but it has to know where to start. So there's another enzyme that comes in and puts a marker, puts a little bit of RNA in it, and there it says, hey, polymerase, come in and copy. So polymerase comes in and goes, copy. Yes? Are you with me? The other strand, not so easy to do because it's in the opposite direction. It's going backwards. So therefore, a new enzyme has to come in and it has to cut, 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 and then primase comes in, puts a marker in each of the pieces, then polymerase comes in, copies, 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 and now you have a bunch of pieces, right? Ligase comes in, glue, 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 and then you have two strands of DNA.
Two whole strands.
Good?
So look, you're at the air.
What did the helicase and the topir?
Helicase opens up.
Helicase opens up,
Mhm.
helicase does what?
Well, let's do a little quizzy poo right now. How about it?
Oh, God.
Topoisomerase, what does it do?
It opens up.
No. Topoisomerase does
Relax.
relax. It's tightly wound, it's gonna open, it's stressed, it doesn't wanna open. Topoisomerase says, open. Helicase does what? Opens it up, forms that Y. Yep. So now, in order for it to be copied, the enzyme that does the copying has to know where to start. What tells it where to start? Primase. So primase puts a primer, it's a little piece of RNA. It basically says start here. Yep. Now what comes in does the copying? Polymerase. Now there's different forms of polymerase. There's pol 1 and pol 3. I don't need you to know the difference. I just need you to know polymerase does a copy. Okay? Everybody with me? So that's in the leading strand, and we believe that the nucleotides get copied approximately 1,000 per second. Isn't that cool? But on the other strand, things are not so simple because it's in the opposite direction. Yes, ma'am, Ms. Valencia?
And you said this is for the lagging strand?
No, this is the leading strand.
This is the leading.
Now, the lagging strand is in the opposite configuration. So it has to be, sometimes it can be pulled up and then cut, pulled up and then cut, and all those things get copied, and then it gets all put back together again. Yes? Those strands or those fragments, those loops, those pieces have a very famous name from the individual who first conceptualized how it happened. Does anybody know what the name of that very famous scientist is or was? Huh? No?
Okazaki?
Okazaki. Okazaki. So they're called Okazaki fragments.
Who said that? Tramp? Tramp said that? Tramp said
You're
that.
hitting
It was somebody over here. I think it was Tram.
She had amino acids in there.
1 point for Tramp.
Tram's got too many points already. You can't give her another. Okay? So, everybody with me? Okay, so sometimes having one short guy in the front of the class talking about things isn't enough. I'm going to show you my favorite video. Are you ready?
Can you say one more thing about?
Yes? What would you like me to say?
So ligase is the glue.
Right.
Nuclease is the chopper.
Molecular scissors.
Molecular scissors.
Yeah,
What's the nucleus do?
the nuclease.
And then.
The nuclease cuts. Okay? So you're not going to see all of the enzymes here, but you're going to really get to see the mechanism, and I really want you to understand the mechanism.
And what does the ligase do?
It puts things back together.
Puts things back together, so we
That's
don't.
the
Like the glue.
It's the glue.
Primase puts an RNA marker.
And then this is for the
Okay.
lagging strand.
That's correct. So endonucleases and ligases are only found on the lagging strand. Okay? Hey, folks, enjoy this. It's a beautiful piece of biochemistry.
Using computer animation based on the latest research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into two strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is two new DNA molecules.
Yes, Ted?
So the bottom one was the lagging strand, and the one that said it has to be copied backwards was the leading strand?
Opposite.
Opposite.
The one that's being continuously copied is the leading strand.
It was getting
The
separated.
one that
and
gets
pulled.
copied, pulled in loops, that's the lagging strand.
Okay.
And I don't know, can you show that again? Basically, the
Absolutely.
top one goes backwards.
I'm
like-
always interested in students who want to see something again.
Sorry, I thought I.
Here we go.
Just pay attention to the.
Using
upper
computer
one.
animation based on the latest research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly
All
line
these
of
little things flying
amazing
around, those are
miniature
enzymes.
biochemical machines that are pulling apart the DNA
So the
double
bottom
helix
is the.
and cranking out a
lagging.
copy of
Leading.
each
Leading,
strand.
sorry.
One direction, very
The
simple.
DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into two strands. One strand is copied continuously and can be seen spooling off to the right.
I see now.
Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is two new DNA molecules.
Could you share this with us?
I'll be happy to do that.
Professor.
It is, however, it is in the PowerPoint.
Is it?
Mm-hmm. So.
If you're a college student like me, especially, you need Grammarly in your life. Grammarly is a digital writing assistant, and it's super easy.
I think I see it on number 68.
It's right here,
Oh.
44,
That's it. I see it.
okay?
Okay, okay.
But,
Yes.
so she says the end result is 2. She talking about from the lagging? When she's speaking of that, or is she talking about entirely, the end result, the product is 2?
The end result of the entire process is 2 strands.
Okay.
So, but those 2 strands are gonna end up being 2 DNA molecules, right? Because if you think about the leading and the lagging strand, both of those become the template for the production of the nucleotides themselves,
Say it
right?
again?
So you have the strand opening up like this, and the both of those strands are gonna just serve as the pattern, if you will, the template for 2 new DNA molecules, right? So the leading strand gets copied continuously, the lagging one gets copied in pieces.
Cuts.
But both of those are gonna be whole new DNA molecules,
Okay.
and those are gonna be in 2 different cells.
That's why when you see the pictures, it always shows 2 daughter cells.
That's correct.
Okay.
That's
Can
correct.
you explain a little bit why one of them goes, like through a complicated process versus the other one that just?
Yep.
I know that it's backwards and it's more complicated, but why?
Okay. Polymerase has a limitation. What does polymerase do? Copies. So on the leading strand, open the leading strand, it's in the correct configuration. Therefore, it can go this way. Yep. Because it's 5' to 3'. The other guy is in the opposite configuration. So because of that, it has to be cut. And then it has to be, instead of being copied this way, it gets copied this way, it gets copied this way, and then it gets put back together again. Because the limitation for polymerase, it can only add nucleotides to the 3' end.
Okay.
Okay? Yep.
So it's because they're going in opposite directions when they're connected, once they break apart, it has to start over on the other end, so it's still doing the same thing, but it's on the other end.
Except the leading strand is going forward, it's going 5' to 3', but the other one is 3' to 5', right? So therefore, it has to be copied 5' to 3', copied 5', and then put back together.
Okay,
Okay.
so the lagging is going 5 to 3?
The lagging is going
3 to 5.
3 to 5.
Okay.
Good? Okay, this is the hardest part of the entire lecture, right here. I want you to be able to tell me, if you look at this DNA molecule, which are the nucleotides that are at play, right? So let's do the hard one. If you look here, what is the base pairing here? Huh? What is the base pairing there? GC, why do you say GC?
Because it's not AT, which is clearly labeled.
Wrong answer. That's 3 hydrogen bonds, 1, 2, 3. Good? So you know that if it's G and C, that a purine and a pyrimidine have to be put together, yes? What is a purine?
Has 2.
2 rings, and a pyrimidine has only 1 nitrogenous ring. Therefore, if this is held together by 3 hydrogen bonds, and that is a double nitrogenous ring, what is the nucleotide right there?
Right where?
What did he ask?
What was it?
2. Wait, no, is it?
Guanine. Why? Because guanine and cytosine are together, and they're held together by 3 nitrogenous rings, and the guanine has got 2 rings. So then what is this thing? Cytosine. It's got 1 ring. Let's do another. What about this one right here? How many hydrogen? 2. So therefore, this has to be what pairing? A and T. So what is this one then, right here? Thymine. Why do you say that? It has one ring, right? And the other one has to be what? Adenine.
What do you mean by
I
one
like
ring?
The carbon rings.
Remember
The
carbon?
nitrogenous.
what the ring.
So it's the,
So-
the kind of the pentagon looking thing.
Uh-huh.
So that's one ring.
Oh,
When you have two, that's two
hello.
rings.
Yeah, right here.
So did you get it, Ellie?
Yeah.
Okay.
Hearing.
Anybody else got questions? So how do I know that this is the 3' end? Yes, hydroxyl. How do I know that that's the 3' end? How do I know it's a 5' end? But why is this hard? Why is this hard?
True. Solid answer. One
easy.
point.
I'm just very.
All it takes is one short little dude to stand in front of the class and tell you what it is. And you should know. Okay?
So the purines always have the double.
Double nitrogenous ring.
Goes back to this.
Pyrimidine 1. Good?
Purview.
Yeah.
How do you know which one, like on the example that you just gave us? How would you know if? Of course, we would know like a pyrene versus a pyrimidine, but like, how would you know if it's adenine or guanine?
Yeah, can you go over
So,
that
yeah,
again?
okay. So first, this is the way you need to do it. As I'm sitting here with y'all, I'm a student. I'm sitting here with y'all, and I'm thinking, hopefully like you are. I'm looking at this, and so therefore I think, oh, I'm looking at 3 hydrogen bonds. The 3 hydrogen bonds hold together the cytosine and the guanine base pairing.
All the time.
All the time.
Okay.
Yes.
Every single time. Therefore, if I'm looking at this, one of them has to be cytosine and one of them has to be guanine. I know that my purine, which has 2 nitrogenous rings, is guanine. So if I look here and I see 1, 2 nitrogenous rings, that has to be guanine. That must be cytosine there because it has 1 nitrogenous ring and it's a pyrimidine.
Gotcha.
That's how you think about this.
I got it.
And the uracil doesn't make a difference to the dynamic at all?
It doesn't at all. It's still held together by 2 high. The only dynamic that is that, and we're going to cover this in just a second, is that RNA is made from DNA. Okay? And RNA is single-stranded. Okay?
Right, that's clearly DNA.
Right. Okay?
So
Good?
adenine has double bond.
Which one?
Adenine?
Yes. Adenine, thymine, not double bonds, but double hydrogen, or double hydrogen bonds, yes. Okay?
And then you said that was adenine and
Thymine.
and
So if you, a good way to remember this is to draw this on your paper.
Oh, I just saw, I just clicked it. I did, but right there. Got it. Thank you.
Okay?
You got it.
Good?
Yeah.
Why is this hard?
Because if you miss one
Why
thing,
is this hard? Okay? Alright. So, I do like to use figures, and I like this one a lot. Can you tell me what the enzymes are here? Okay, I'm gonna point at them, you tell me, okay? Okay, what's going on at number 1?
So please summarize.
Huh?
However you say it, the first one.
Relaxation. So the DNA strand is being relaxed. What enzyme does that? Topoisomerase. Number 2 is the one that unravels and unzips the molecule. What does that do?
Helicase.
Helicase. Now, if you look at number...
That was 2 out of 2 wrong.
If you look at number 5 is putting a little piece of something or another to basically say start here.
Primer?
That little pink thing is a primer, but number 5 is the enzyme called primase. Number 6 is doing the copying.
Polymerase.
Polymerase.
Oh my god.
Thank you for that.
Thank you for not testing us on how we say things.
What did you call it?
Oh.
Polymerase?
I'm okay with that.
Polymerase
I'm
or
okay with
something.
that. I'm okay with that. Polymerase,
Polymerase. Say the T one again?
topoisomerase.
Topoisomerase.
That's number one, topoisomerase is one.
Topoisomerase.
Okay, now, there is no nuclease here. But number 7 is putting them back together again. What is that? Ligase. Ladies and gentlemen, what's Christian today? Ladies and
You said
Eric.
you'd be late or maybe not make it at all.
Oh, okay.
So you send something in the group chat.
Okay. Here is the answers. It's in the PowerPoint.
I do like that you said primer. I like those things.
Right?
You probably should
Oh,
know.
we don't have it.
You don't?
We don't have it.
Oh, I might have done it like late at night one night and I forgot to put it up.
You got a blank one.
Well, I tell you what, I'll send it to you. How about that?
Thank you.
I got a lot of things to send you. You guys?
What's the difference between topoisomerase 2 and topoisomerase 4?
Just the way they interact and where they interact.
Okay.
So I just need you to know topoisomerase, what it does.
Topoisomerase.
If you become a biochemist, then you and I can talk about T2 and T4.
Thinking about it.
Okay. Yes?
Can you just say like one sentence about the Okazaki?
Okazaki fragments? Okazaki fragments are the pieces of the lagging strand that are copied and then put back
So
together.
like the fitting piece fragments.
Correct. The pieces that are copied. The important part of the DNA molecule. Okay? This is DNA replication. We haven't even done protein synthesis yet. Okay?
Mm.
Everybody with me so far?
Mhm.
All right? Yeah.
3 and 8's on there?
3 and 8. So 8 is just a Okazaki fragment. It's just a piece. And 3 I did not talk about at all. 3 are small binding proteins. They add stability to the DNA molecule when it's being replicated. I will not ask you about that. Okay? Thank you for pointing out, because sometimes I forget. And if we were in my 1406 class, I would be talking about those. Here I'm not.
And what is 8 again?
What is what?
Okazaki
Do you
fragment.
really want to know? I'm not going to ask you, but yeah, it's an Okazaki.
What's the 14 and 6 class?
It's cellular molecular.
Ooh.
It's majors biology, first semester. I teach that on Monday, Wednesdays. Come visit.
Can we just come in and sit in there?
Huh?
Could we technically just come in and sit and listen?
You could come in and visit. I would be okay with that if you want.
Is it what? What's the class?
Cellular molecular.
Is that what you guys are talking about right now?
We're talking, you know what we're talking about right now? Cellular respiration.
We know that.
And I don't really deviate as much. I give my full cellular respiration lecture to you all. I just do more with enzymes with them than I do with you.
I appreciate you not doing that.
Okay. Are you ready to move on? Let's do, there's the video. Let's talk about protein synthesis. So protein synthesis, remember we have 3 different types of messenger RNA. We already talked, 3 types of RNA. We talked about messenger RNA, transfer RNA, and ribosome RNA, okay? Remember, they have different functions. Tell me what the functionality of messenger RNA is. What?
DNA gene information.
Copies DNA into RNA language, right? What's the purpose of transfer RNA, tRNA? Brings an amino acid to the growing sequence of amino acids, right?
What codons say and how to put those into action?
That is correct. So codon is in the messenger RNA. There is a complementary 3 nucleotide structure that's found on the transfer RNA. And guess what it's called, Samantha? If it's called the codon on the messenger RNA, what do you think it's called on the transfer RNA?
Anticodon?
The anticodon. The anticodon. But they pair up with each other, okay? And then the ribosomal RNA folds a protein. Yes? Good? So, let's talk about, this is eukaryotic, so just listen. Just listen. So it's all happening at random. This is all happening at random. Okay? So here you have the nucleus, and so there's the DNA molecule, and this thing is copying the DNA molecule in RNA language. Here it is when it's finished. And it leaves the nucleus through a nuclear pore. What is this thing called? mRNA. mRNA comes down to the ribosome and makes a complex. It forms a complex with it. And so 3 of these nucleotides is called what? A codon. And it codes for what? An amino acid. And the reason that transfer RNA knows which amino acid to put in the sequence is because it has an anticodon associated with it. So whatever the codon reads, there's going to be a complementary anticodon, and that's going to basically call for an amino acid. Yes?
So it's primase?
No. Mm-mm. This is at the formation of a protein. Primase is mostly talked about when we talk about DNA replication. Do not confuse DNA replication with protein synthesis. They're 2 different processes.
I will.
Don't do it. Okay? So here you can see, that particular complex, right? And everything that's going on. These little bitty things are representing amino acids. So little things look like firecrackers represent transfer RNA. They're picking up the amino acids and they're bringing them in, and where that codon reads and is complementary to the anticodon, that's where that transfer RNA is attaching, and then you can see the chain of amino acids growing. Okay? The other one.
Okay, can I just sum that up real quick?
Yes.
So in the nucleus, we have DNA replication.
Yeah.
That mRNA.
Time. But DNA replication is different than the production or the copying of a segment of the DNA by RNA. So DNA replication is making one DNA molecule into 2. We just talked about that with all those enzymes. Here, what you're looking at is the copying of the DNA molecule into an RNA segment.
Yes.
Okay?
That's
And
called
that-
protein synthesis.
No, that is called transcription.
Transcription, yes, you're right.
Okay.
So DNA transcription happens in the nucleus. The little piece, the mRNA, gets out of the pore into the cytoplasm where the ribosome is waiting for it initially. The ribosome is the translation part that has anticodons that.
Transfer RNA does that.
Oh, yes, sorry, the transfer RNA, which is what we can.
The little firecrackers.
Is, yeah, in the cytoplasm. So they go to the ribosome, they're in the ribosome eventually, and.
They attach to the codon on the messenger RNA. And on the very bottom of the transfer RNA is an anticodon, which is complementary to the codon on the messenger RNA. What we mean by that is if there's a C on the codon, there's going to be a G on the anticodon.
So you said the anticodon.
We say that that's complementary, that they fit the base pairing. Yes, ma'am?
The anticodon is on the bottom of the amino acid?
The bottom of the transfer RNA.
Transfer RNA.
So Tay, the amino acid is attached to the very top of the transfer RNA.
Okay.
Okay?
Is that an image you really, really, really like too?
No, I'm not gonna show it to you on the exam.
But we need to know that.
No. You do not need to, I'm not gonna show you this on the exam, I
But we
promise.
need to know the process.
The process is different, and I'm gonna talk about the process right now.
Can I have
So-
a cookie?
Yes, you may have a cookie. As a matter of fact,
think my
anybody else want a
brain
cookie?
is running
It's
out
good
of
brain
glucose.
food right now. Cookie, just give a cookie.
I'm taking 2.
Give one to Jess, she's really wanting a cookie.
One
All right,
Here,
so
I'll pass it
if
around.
we think about the process of just protein synthesis, this is after the DNA has been copied by messenger RNA. Messenger RNA has left the area where the nuclear material is, and it's now complex with the ribosome. This is where this process happens.
That's translation.
Not yet. So we've only had transcription, but now it's all in one complex. Yes? We're about to undergo translation. Translation really takes part in 3 different phases. Initiation, start, elongation, the continuing of the adding of the amino acids based on the number of codons that are in the messenger RNA transcript. And then termination. We get to a termination sequence that doesn't code for an amino acid, it just codes for stop. And that means that the process is complete. Oh, good, we've done really well. Anybody else? Cookie? Cookie?
Thank you, these are great.
They are,
Yeah.
They're so good.
Okay, so let's go. So here you can see is, what is this thing called right here? mRNA. This is called the large subunit, that's called the small subunit. There's an E site, a P site, and an A site in the ribosome. The E site stands for exit, that's where transfer RNA leaves. There's a P site, it's called peptidal. The peptidal site, what does peptidal mean? Like in the peptidoglycan, put into a chain, right? The peptidal site is where the amino acids get put into a chain. And the A site is the acceptor. This is where the transfer RNA is accepted by the messenger RNA. Yes? Everybody with me?
What was that?
Is the acceptor. What's that, Jin?
Are we expected to know what E, P, and A means?
Would you like to?
It's just a lot right now.
Okay. Maybe.
It is a lot.
I'm just
Trying to keep up.
Yeah.
It's all genetics.
It's not all genetic. This is just very basic DNA and RNA biology.
Oh.
You'll be all right. There have been thousands of students who've become before you with less aptitude than you have, and they've done just fine. Why would I expect you not to be fine?
I'm just
Yes,
like,
You said the A is acceptor,
Acceptor.
but transfer RNA to what?
That's where transfer RNA is accepted by the messenger RNA. Okay, yes?
So the A site is where initiation happens?
No, okay. Yes, but we don't say that, Tram. What we say is we say that the initiation sequence always happens in the messenger RNA, but it's always going to start at the A site. But what we do say, Tram, is that initiation of protein synthesis always begins with the codon AUG.
Why is it AUG?
Because that's just the way it is. That if we look at all of the research that's ever done, anytime we say AUG, that's the start. The Brits, when they first decided this, said there's something very formal about this AUG, and they called it the formal site all the time.
Okay, so if you were to ask us what is the starting codon?
You'd say?
AUG.
Correct.
Or if you said, what's the formal site, you would say.
I wouldn't do that. I would more likely say, what's the initiation site, or what's the starting codon, something like that.
We can
Okay.
remember it by saying, and go, AG.
Okay. Okay. So the initiation, the start codon always codes for methionine. Okay? So if you look at, did everybody get a codon table? Everybody got a codon table?
Yes.
Oh, you didn't get a codon table?
That makes sense.
Hey, give one to our buddy over there.
I have a question.
Yes?
What is, can you repeat what E is?
It's, the E is the exit site.
If you quickly remember them because school starts in August.
So synthesis starts.
I
in
don't wanna
August.
get into any of your little sayings because they don't make any sense to me.
Does that make sense though?
Okay, if it makes sense to you all, you all just stay with it.
Hey, we thought we had something here.
So, okay, so this is the initiation, always starts with what, Eric?
Methionine.
AUG, very good. Elongation is just a continuation of the placing of the amino acids in a chain due to what is in the codon of the messenger RNA, right? And that'll continue, Ms. Valencia, until a termination sequence hits the A site. At the A site, if you have one of these termination sequences, UAA, UGA, or UAG, that just means stop. It doesn't code for an amino acid. It just says the process is finished. And remember, some of these proteins can be thousands of amino acids long. Okay?
How long does that take? Are those things just milliseconds?
Seconds.
It's cranking out, and it's
Yeah.
continuous.
Continuous. And in bacteria, they can do polyribosomes. They can have ribosomes and chains, and the messenger RNA comes in, gets in one, gets in another, and so all of a sudden you have all these proteins all at once. Well, it doesn't happen like that in eukaryotic cells. Sometimes simple is much better than complex. It does take energy.
So that's why DNA can replicate so fast?
Yeah, so we believe that DNA gets replicated at 1,000 nucleotides per second. Yes?
So the termination codons, do they happen like randomly or like on purpose?
No, no, they don't happen at random. Well, they might happen at random, but that means the protein will never work. So usually it's at the end of the sequence, and then that means the protein is done.
Oh, I mean like when it ends, like does it ends like randomly or like
No, it ends at the very end of the protein. Now, Tram, it can, in some cases, be put into the middle of the configuration of the protein, but then that protein will never function. It won't never be made. Okay?
Okay.
Yes.
Do the different stopping codes, do they kind of decide on what the different proteins will be?
No.
So different cells have all of the DNA sequences in them, but only certain parts of the sequences are turned on, and they only make those certain parts of the DNA into the protein synthesis.
And that's why your tongue is your tongue.
Mhm.
And there's no toe in your tongue.
Okay.
You make it seem so simple, I'm so confused.
This, I know, guys, listen, I understand that this is abstract. I understand that a lot of people need visual stimulation to see this. It's really hard for somebody who's an auditory learner like myself, who can listen to things one time and understand it, and other individuals are more visual, they're more tactile, they're more kinesthetic. And so therefore, I have a really nice video for you to watch. Would you like to watch it?
Yes.
I knew you were gonna say yes. Amy, this was for you.
That's what I'm gonna do when I get home anyway.
Are
Watch
you gonna do
different
the
videos
Aviva
until I
sisters?
understand.
What's that?
Are you gonna watch Aviva Sisters?
Do you have is anybody recording this lecture?
Hey, you know, this lecture is recorded on my YouTube website. It's there.
It is?
Yeah.
What's the name of that?
DNA and RNA biology.
Okay, awesome.
Every single lecture he did while he was online is on there.
Yeah, I wasn't smart enough to get them all, but I got most of them.
I got really into it doing my research yesterday. and
Yeah, I
was
can
just-
tell, 6 pages later, only done with one, right, so. Okay, listen up. Now, this guy is a lot more monotone. He doesn't get as excited as I do. But he will tell the story nicely. This is in bacteria. Here you go.
Translation is initiated by formation of an initiation complex consisting of the 30S ribosomal
Small
subunit,
subunit.
formylmethionine tRNA, and messenger RNA. The 50S ribosomal
Large subunit.
subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has two sites to which transfer RNA carrying amino acids can bind. One is called the peptidyl or P site, and the other is called the acceptor or A site. There is also a third site called the exit or E site, where transfer RNAs are released. The initiating transfer RNA carrying formylmethionine binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The formal methionine carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site by a peptide bond. The ribosome now advances a distance of 1 codon, and the transfer RNA that carried the formal methionine is released at the E site. A transfer RNA carrying the next amino acid now moves into the A site, where the anticodon on the transfer RNA matches the codon on the messenger RNA. The ribosome shifts down by a distance of 1 codon. As the shift occurs, the 2 amino acids on the transfer RNA in the P site are transferred to the new amino acid, and the second transfer RNA is released from the E site. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when a stop codon moves into the A site. A stop codon does not specify an amino acid and does not have a corresponding transfer RNA. The ribosome dissociates into the 30S and 50S subunits, and the messenger RNA and protein are released.
Now your picture makes a lot more sense, the one with the polysynthesis in detail.
I try my best to make sure that I try to reach everybody who has a different modality of learning. So I will always try to make it so, this particular video that we're not gonna watch here is a HHMI, it's one of the better ones, but it shows the folding of the protein. I'm not gonna talk about that, I'm not gonna test you over it, but if you wanna understand a little bit more, Miss Isabella, then you can look at that if you want. Yes, sir.
So he kept on saying that the anticodon will get released, but I thought
The anticodon is part of the transfer RNA. So the transfer RNA gets released, and so does an anticodon goes with it.
So is that just I thought it stays in a chain initially.
Mhm.
So
So what stays in
broken
the chain
into-
is the amino acid.
I'm sorry, so, cause that, yeah, exactly, so.
So here is the chains of amino acid. There is a transfer RNA, that little blue little thing down there, that's the anticodon.
Yes, so the anticodons are getting
Released.
reused?
Uh-huh, over and over again. Okay?
So that firecracker thing, what's that again?
The transfer RNA.
Transfer RNA, so it goes back out and looks for that protein, and it always
No,
has
amino acid.
amino acid, and it always has the same anticodon on it.
Correct.
Okay.
Yes. Valencia.
Oh, no, that makes sense now.
Okay. All right, so, wait. How do we know which amino acids are being put in the chain? Because we've done a lot of research, right? And so this is your codon table. Everybody's got one. The way you read these things is this is the first codon, there's a second, and there's a third, right?
What, do that again, I'm sorry.
First codon,
Uh-huh.
first nucleotide, second nucleotide, and third nucleotide.
So
So
like
if you look at this, let's just practice. What is coded by CCU?
GGU or CC? Like,
Oh, I'm sorry, GGU? Glycine. Right? What is coded by ACC? Don't say Austin Community College.
Threonine?
Threonine. And what is coded by TAG?
None.
Nothing,
Why?
because it's not on here.
Huh?
Because thymine is not in RNA.
Thymine is in,
or DNA.
no, DNA. Thymine is on
Oh,
DNA.
this is RNA.
This is RNA. Right, so it can never have T's.
Codons are only RNA, yeah?
Correct. Good?
Code,
All right,
wait, say that
now-
one more time, Eric?
Codon is only RNA.
That's only RNA.
Thank you.
Now, here is a transcript. That transcript, can you give me the complementary messenger RNA? So this is DNA, Can you give me the transcript, the messenger RNA?
A, G, G, C, T.
Wait.
G, or
Is
C.
you. C. Is it you?
C.
T.
A.
Oh, so that's
There
D.
shouldn't be a T then, no?
No.
That's right, U.
Oh.
You.
A.
A.
G.
C.
What?
C.
G.
Oh, G.
U,
A.
T.
T.
U.
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 Oh, there's no T.
Okay. Now, how many codons are here?
1,
Five.
2, 3, 4, 5.
Good. What does AUG code for?
Start.
Start.
Which is?
A.
Initiation.
Methionine.
Methionine. What does CUA code for?
Leucine.
Loosen.
Lucy.
Leucine.
Oh, okay.
Lu.
What about GGA?
Glycine.
Glycine.
What about AUU?
Isoleucine.
And what about UGA?
That's a stop.
Will you be able to do that for me on an exam?
So this is
No,
the
we
first
just code our
letter,
table.
middle letter, third
With
letter.
studying I
No.
will.
What's that?
With studying I will.
Oh, you
Will we have to go
provide
on
a table for
I
us?
won't provide the table.
Oh, thank you.
You do not need to memorize the table.
This is like,
That's just crazy.
yeah, it's like, you're gonna.
That's what you guys need.
Wait,
Wait, what?
I wanted to take a picture.
But it's all there on the PowerPoint.
Remember this?
You want to take a picture now?
No, it's okay.
So that's what it's going based on.
Good?
So the whole purpose of RNA, well, DNA to mRNA is to be protein that the body can use to synthesize.
Cells.
Cells.
Yep. Good?
No.
So people always ask me, Proby, why are you putting us in this torture? It's so important to understand this because as you're gonna find out, the mechanisms of the 3 antibiotics that I just gave you, one of them is gonna work on RNA, one of them is gonna work on DNA.
Cause doesn't it target like the 50S and I was looking at.
So it's important to understand this. Plus, it's on the registry. It's on the actual NCLEX and the one for dental hygienists also.
So
Can you say that again?
So basically,
Sorry.
no, I was just trying to connect it with the antibiotics. So with the antibiotics, it's stopping it once it hits like the ribosome, right? Cause that's like the 50 and so.
If it's stopping at the ribosome, there are no proteins.
And the only thing that changes is
What were you saying that it's on the NCLEX and on the dental hygienist?
On the exams.
This subject matter?
Yeah.
Okay.
makes sense.
Good. One more thing I need to talk to you about is mutations. And I know I'm a little bit over, but lab is so short today. So, might as well get it done. It's not a lot. Jen, are you okay? You're making faces.
Sorry, it's just not, I haven't had a good day. It's
You
okay.
sure it wasn't the face of boundless enthusiasm?
No, it was like this.
Oh, really? That bad?
So, mutations, write this down, mutation, any inheritable change in the DNA molecule.
Any and.
Heritable change in the DNA
How do you spell
molecule.
inheritable?
Inheritable?
Any inheritable change. In DNA molecule?
Mm-hmm.
In DNA molecule.
Okay. So there are lots of things that can cause mutations right now. If we look at bacteria, mutations in bacteria are always good for them because it changes the way that they survive. It always gives them a leg up, an advantage. Mutations for us, not normally are they good. Usually they're bad. And so mutations for us can be changes mostly in our somatic cells, in our body cells. And we, unlike the bacteria who have no way to find them and correct them, our cells have ways to correct them. And so we have checkpoints throughout the process of the DNA being replicated and being distributed. And any of those checkpoints find anything wrong, they can repair. And if they can't repair, then a signal is sent to the cell, and the signal says what? Die. That's programmable death. It's called apoptosis. Okay? Sometimes
So
the signal,
apoptosis is programmable.
programmed cellular death. Sometimes the checkpoints don't work and the signals don't work. And therefore, we develop these cells that are growing abnormal. The cells don't know they're abnormal. They just do what they do, right? And our immune system looks at them and says, you look funny, but you have the markers that say you belong here. We're not gonna destroy you. And then you have cancer. So cancer can be in our bodies, and statistically, in this cohort of people right now, 3 of us have cancer cells in our body already.
I guess we're all making numbers like
Well, that's why screening is really important, right? So the most aggressive forms of cancer are breast cancer, right? Very aggressive, and then testicular cancer, prostate cancer, fairly aggressive. So that's why we want to do these. We want to do these screens throughout the milestones of life, which I just hit one. So now I have to go get colonoscopy.
I mean, I hear people say all the time, like, blue eyes are a mutation and
No.
red hair is a mutation.
So they're not mutations, they're part of the genetic makeup of people. And so they are traits that are transferred to progeny. And statistically, there are just not that many redheads, so to me, they're pretty rare and pretty cool. And usually redheads have freckles and blue eyes, and so they're just laden with all kinds of recessive traits.
And their
right?
hair never goes gray.
We turn white.
Well, okay, I don't know that.
It is true, our hair will turn white, it isn't gray. I have white pieces of hair.
Okay. I didn't know.
It goes bright white.
Yes.
I have a question, to the markers that you were talking about. So those are markers that indicate that they belong.
Every single cell in your body, with the exception of your red blood cells, are marked with your markers.
So a follow-up question, my dad actually got diagnosed with prostate cancer last year, and they found it because he got blood work done. Are those the same markers that we're talking about?
So I don't know if they might have done a C-reactive protein to find that. I don't know how they found it. It could have been an indicator in the blood, but they could have also done a C-reactive, and the C-reactive protein might have been higher, and then they would have started checking other places and would have found the prostate cancer, right? There's lots of hints that there's something going wrong with the body.
Yeah, he didn't have any symptoms, the doctor.
So
or at
you,
least
unfortunately-
noticeable symptoms.
Unfortunately, when we as humans start to show symptomology of cancer, the cancer's progressed to stage 3 or stage 4.
Well, that was kind of an interesting thing though too, because like the doctor was just, like you said, like, yeah, you should get a checkup because you're hitting the milestone. And that's exactly what he did, and it was during COVID times, kind of still when it started, so they kind of pushed it a little bit and he couldn't go to the doctor all the time, so every like 3 months they checked his blood and it got pushed a little bit further, and eventually they saw that, I guess, the marker that they talked that they were
C-reactive
looking
protein.
for
It was probably
was
the default.
elevated, and then they went and did a biopsy and it turned out to be.
But it could have also been prostate antigen. So there's a lot of ways you could figure out what that's going on.
But yeah, that's why I really want to make a point to like get checked more.
One of my good friends who teaches here,
checked.
she was gonna have a hysterectomy, and she went in for her last checkup, and they did a X-ray of the area, And we said, we decided not to have a hysterectomy, but we want your permission to go in there and remove this mass that's on your kidney. And she had no idea. And so she and her husband talked about it, they said yes. And so when they were in there, they found that the cancer had completely engulfed the kidney, so they just took the whole kidney out. But had she waited another year, the cancer would have metastasized further. So serendipity, by luck, she was gonna have a hysterectomy, and they saved her life because they were doing a last checkup before the hysterectomy surgery.
I hear lots of stories like that, some that don't end as well as you want them to, because it's too far progressed, but my mom, she had colon cancer and she didn't know it.
Her dad.
And she went in for a hysterectomy, a colonoscopy because she has turned 50 and all this stuff, and they found it and they were able to take it out before it got
My
heavy.
dad didn't do any of those screenings. And so I remember one Thanksgiving, I was visiting with him and my mom, and he said, I keep burping, I don't know what's wrong with me. I said, it sounds like you have reflux disease, Dad, you need to go get it checked out. Well, he waited another year. When he went to get checked out, he had adenocarcinoma of the stomach and he went on stage 4, so he didn't survive. But, you know, had he gone back then, if I had pushed him, you know, well, I don't live in Corpus
But you
anymore.
know how
so-
men are.
Exactly.
Not to be a downer, but it's also like really important, like for young people as well. Like I'm 28, and my best friend 2 years ago got diagnosed in COVID with testicular cancer, and he put it off for a really long time, and it was right around the holidays, and they couldn't do a scan. Like I don't know what happened because of the holidays and COVID, they didn't do a major scan,
Well,
and it had
they
spread.
weren't doing anything during COVID
Mhm.
at all that allowed for people to go into
It was
an
really
outpatient
tough.
facility.
Yeah, well, he was getting scanned. I mean, it had spread to his limbs, that had spread, but like because around it was like on the 24th. We found out on like the 20th that he had cancer. On the 24th, he was gonna go see if it spread. They didn't run the scan, it had spread. But he had like found a lump and was like, oh, I just don't know. Like, and then it got to the point where he couldn't walk. And this was when he was 26. But then I got scared and I had found a lump, so then I went and it was just benign, but it's like anything can happen.
One of the scariest things that ever happened to my wife and I is we found a lump on her breast.
Yeah.
And I thought, oh my God, this, you know, so you can feel them. and
Yeah.
If you move them, you know.
It's just more hard.
And I was like, oh, this does not look good. But it came back benign, and it was just a cyst.
But it's just so scary.
It scared the hell out of us.
Yeah, I feel like cancer is something where it's like you don't want to go there. Like, oh, I don't want to get that in my brain, but it's like go and get checked.
My
Just go get
rule of
checked.
cancer is if you can cut it out, you should.
Yeah. Oh, 1,000%.
But not everybody believes that. People believe that they don't want to have surgery, they'd rather fight it with radiation and chemotherapeutic drugs. Cut it out. Because if you cut it out and you get it off, it's not going to hurt you. Whereas if you leave one cell because of the radiation or the chemotherapeutic drug, one cell, it'll come out.
I think that's one of the good things about the amount of a platform somebody can have online now is so great, because it's a lot of people, like there has been famous people and things like that they've put that on their platform, so it enables more people to get checked, and
The most
things
famous
are caught
person that
earlier.
ever said anything that I thought would help somebody was Angelina Jolie.
Oh.
yeah, 'cause she had
Angelina
like
Jolie
breast
had a double radial breast mastectomy. She had no cancer, but she had the BRCA gene.
I had no idea.
And because of that, she decided to remove her breast. So
Because it's
that-
like
A lot of
really, really
women
high
do
chance.
that. Yeah.
Because her mom had been really sick with it, and maybe I don't know, but her mom had been, so she
Wow.
decided to remove them. Now, Angelina Jolie has a lot of money.
Yeah.
But not everybody can do that and then have cosmetic surgery to make your body look really great.
That is something that's really tough. Like, I don't have insurance, but we have to pay out of pocket every single year because cervical cancer. My great grandmother had cervical cancer, and so I pay the money to get checked every year.
Yeah, you should. What I will tell you that's, we wanna go from a downer to an upper.
Yeah.
Is that if you look at the latest statistics for nursing, we were down 200,000 before the pandemic, we're now down over 800,000. There's so many nursing jobs. And dental hygienists can write their ticket anywhere they wanna go.
Yeah, they're
They're
in
in
need
such
right
high
now.
demand
Oh, yeah,
right
they
now,
are in need.
that a lot of them are just quitting their jobs, working 1 week a month, and just playing for the other 3 weeks because they can make that much
I
money.
We
told my
were
husband
paying
when I
one
graduate, I'm not
She
working
makes
full-time.
75 an hour.
We were paying
She
dental
gets to say
hygienists
what
temp
she
85
thinks.
an hour. We were paying
It's
them
amazing.
85 an hour.
That's why I'll be temping.
So you guys are here at the right time. Stick with it. Follow your path. Never compare yourself to anybody else. You're on your own path. Do not compare yourself to somebody else. They're on their path.
Just tell the director of the program that we're really smart and to get us in.
I wish I
Yes.
had that power.
Oh my God.
I'll be applying
Oh
in
my
February.
goodness.
Tell
So
her.
I
Tell
wanna
her.
just talk a little bit about how mutations can occur, and so they can occur in nature, because bacteria are the only group of organisms that can take up DNA directly from nature.
Say that again?
Yeah, so bacteria can take up DNA directly from nature. A fish dies, some of its DNA is there, the bacteria can take some of its DNA and incorporate that into its own
Are those
genome.
plasmids?
Can be, but they don't have to be, okay? So transposons are what we call jumping genes, because they can enter the actual cell through a plasmid or some other mechanism, and then they can be incorporated into the genetic code of bacteria, and then the bacteria is forever changed. Okay? So you know about conjugation and plasmids, and these happen also. And where before I didn't say, because I didn't want to talk about the fact that plasmids could become incorporated into the genetic code, they surely can. And so because of that, we were talking about plasmids in Unit 1, here we are in Unit 2, visiting them again, but now talking about a different mechanism, how they can be incorporated into the genetic code and change that bacterium forever. Remember, bacteria don't undergo sex, so the only way they can change is to mutate, okay? And then the last thing we'll talk about today is how viruses can affect cells. So viruses, when they infect cells, they deliver a nucleic acid to that cell. And in some cases, that nucleic acid, the viral nucleic acid, stays with us forever, right? So we call those types of viruses lysogenic. What that means is that they can remain in our bodies forever, like herpes. Herpes stays at the base of our nerve cells, and when you get stressed out, it erupts on your skin, either in the buccal area or in the urogenital area, right? But also shingles can do that. You can have chickenpox as a very young person. The chickenpox goes dormant for a long time, and as an adult, the shingles pop up, right? And so you can get, it's lysogenic. It goes dormant, and then you get stressed out. And right when you think you're at your lowest, Isabella, the virus says, it's time for me to give you hell, and it will.
Why do they say?
Yep.
So you said stress will cause that to come out? How
No,
stressed
no
do you have to
shingles.
be?
Not very stressed at all, too much homework.
Well, I had chickenpox when I was a kid.
Yeah, but-
I don't have.
But it's
chickenpox.
No,
elderly
and,
or
but
more
it might
older
be in your
people.
body,
And
you're
I
not
get
gonna
stressed
know.
as hell.
Well, so stress can actually be being pregnant. Stress can be getting a sunburn. Those are all stresses to the body. It can be, oh my God, I have a Pro-V and a WADS test in 1 week, right? So watch around midterms, you're gonna see a lot of people walking around with fever blisters. That's herpes 1. Simple as that. Yep. Okay. Anybody have a question?
Can you give another example of a lysogenic?
So those are the two that,
Herpes and shingles or?
those are the two that I always like to talk about.
I was.
HPV can also be one.
Okay.
Yep.
Do you think, I'm just curious, do you think the viruses predated the bacteria, or the bacteria came from
That
viruses?
is a great question.
Did the
I
chicken
think
lay the egg?
that the viruses came after the cell.
You think so?
Because what would they have infected before
Oh, they
that?
would have had to have the cell to
It's a
reproduce
good
them.
thought. Just a self-replicating little portion of DNA out there and says, I can just keep doing this, as long as I have a host.
They gotta have host.
Yeah.
Right.
Can viruses infect bacteria?
Uh-huh. which is pretty cool, because to me, I think we should be using phage therapy, so which is being used in Europe now. So if you have an infection of a bacteria, phage therapy, phage, P-H-A, phage means virus, virus therapy. So if you have a bacterium that we can't get rid of any other way, let's just stick a virus in it that's very specific for that particular bacterium. Won't do anything else to anything else, just that bacterium. It will kill the bacterium and nothing else. And then it'll be, oh, I don't have any more cells to infect, and that's when the immune system says, now you're done, and it destroys it.
That's kind of how antibiotics used to be before there was all of this resistance. Like they
Antibodies still work the same way. Antibiotics is what we develop resistance. We don't, bacteria develop resistance.
But would they be able to develop resistance for viruses, or?
There are some, well, almost all of our antivirals don't work all that great. The one that works beautifully is acyclovir, which is the one that's given for herpes. Okay? Anybody else got questions? That is the end of the material for Exam-E-Poo number 2. Exam-E-Poo number 2 is scheduled for Tuesday. The morning class has already asked to move it to next Thursday. Do you want to do the same thing?
Yes.
Yes.
Do I have a motion on the floor?
I have a question.
Yes.
But that means that every other thing that we do from here on out is gonna be super cramped?
No, it's not.
Okay.
So if I move the exam to next Thursday, I'm just into Unit 3 a lot deeper than I would be. We're not gonna take a break, we're just gonna say, okay, exam's Thursday, we're not doing anything today.
No, I was.
So nothing gets cramped.
Okay.
So we're gonna cover some new material while we're studying. for-
Yeah, so that, I'm still having the review on Thursday because
This Thursday coming up?
that's, yeah.
That's perfect.
That's good.
Because everybody already knows that, I'm not gonna change that, but I will change the test based on you all saying
Thank you.
next
Morning
Thursday.
class.
No, can I like